So our final speaker in this very exciting session is Dr. Roy Sillitoe. He's an associate professor in the Department of Pathology and the Neuroscience Program and the Duncan Neurological Research Institute at the Baylor College of Medicine. And his lab focuses on developmental origins of complex neurological conditions. And he's specifically using the cerebellum, part of the motor system, as a model for understanding brain development and disease. Please welcome Roy.
Thank you very much. It's a tremendous delight to be here and to share some of the work that we've been working on at Baylor and the NRI. So I'm going to tell you a little bit about what we do in terms of trying to understand where motor diseases arise in the brain and potentially how we can track what they do in the brain and how they manifest as behavioral outcomes, and potentially how we can start to think about potential treatments in the future. So we study the cerebellum, which is located in the back of the brain. And initially, when I started out in my training, what I was really interested in was the basic biology in how you wire up this part of the brain. It only accounts for about a tenth of the weight of the brain, but it has more than 50% of the neurons in the entire brain. Since moving to Texas, what we've been interested in is not only how they wire up, but what happens when that process goes wrong. What are the particularly the pediatric neurological outcomes if you don't put together the motor system in the correct way that it should? Why we study the cerebellum? One, it's a very tractable system in terms of its cell types. We know a lot about its anatomy. But in terms of what it actually does, is visually tractable as well. It is responsible for many different aspects of movement, from coordination of the limbs to posture, balance, and learning. I've put up this picture that I drew many years ago of Leonardo da Vinci. I've always been a great admirer of what he's done. And I was reminded to share this with you recently when I took my children to the wonderful exhibit at the George Bush Memorial Museum in College Station. He was at the cornerstone of what it is to be an inventor, an engineer, and a biologist. And if I was so lucky to be transported back to the 1400s and talk to him, I would tell him that what he really wanted to understand was the way the cerebellum works. The cerebellum is responsible for this beautiful intersection of smooth movement and how you execute this movement in the context of every other behavior that you perform at the same time. When this cerebellum goes wrong, the consequences are very obvious. In the lab, we think about them generally as three broad disease groups or phenotypes if it's a mouse. You can have a tremor, which is a very rhythmic shaking of the limbs and the head. You can have an ataxia, which is an inability to coordinate your gait. And most recently, we've been interested in a disease called dystonia. This is where the muscles contract uncontrollably, particularly your agonist and antagonistic muscles co-contract, producing extremely painful outcomes, particularly in children. I say particularly in children here because when you damage the cerebellum during development, you have some of the very same outcomes, but ever more so severe. We're interested in understanding what are the mechanisms that produce that. How do you get a circuit that will produce these different behaviors, a very simple circuit compared to what we've heard this morning, but different outcomes in their severity, whether you're during development or a child or during adult. What's also intriguing is that they don't have to be separate. You can have these devastating outcomes all at the same time. The question is, how does this happen? And how do we disentangle this from an experimental perspective? We start with the cerebellar circuit, which here is a bit of a tangled mess, but I promise you we have a lot of good indications of what these different cells and circuits mean. More than 100 years worth for that matter. The idea behind studying the cerebellum gives us access not only to the synapse, the circuit, the network, but also the system and the behavior, which I would argue you put that together, and that's what you really need to understand to fully appreciate a neurological disease or a neuropsychiatric disease for that matter. We study the mouse cerebellum, and we've heard a lot of the comparisons. I've simplified this a little bit, especially the one on the top, same cells as human. Yes, indeed, there are some specializations for primate and particularly for human as well. But I think fundamentally, there's many of the same cell types. And what we understand from the cerebellum particularly, because it's a very well-conserved structure, we can apply to human disease, especially when we model it in the mouse model, as I've shown here, our model of choice. If I take a slice through the cerebellum, I've already told you and tried to convince you that it's a relatively simple structure. But when you look at the gene expression patterns and what I'm showing you here, the protein expression, you suddenly have a level of complexity that's not apparent to the eye. The proteins are expressed in this beautiful and very intricate map that's conserved from small rodents all the way to human. There are parasagittal stripes throughout the brain. Mice have them, we have them as well. And as a postdoctoral fellow, what I did was try to understand, why is the cerebellum stripy? If stripy is actually a word. I just made one up. It turns out that the proteins that are expressed in these patterns guide axons, or the sensory inputs, into very particular domains. For example, in this case, the ones that are labeled here, pseudo-labeled green, or these proteins, will guide hind limb information into particular domains. The ones immediately adjacent to it will guide forelimb information into those domains. Okay? There's a reason you have this wonderful map of complementarity in the brain. You need parallel processing to execute a movement that is so complex, such as locomotion. But what's even more devastating, or more interesting for that matter, I should say, is what happens if you don't have that parallel processing of this information that's been packaged in a very particular manner. I'm going to talk a little bit about dystonia to get us started. This is a disease, as I mentioned, where the muscles work against each other rather than with each other. It's the third most common movement disease right now. Because it can affect small body parts, in fact, in this case, on the very left is just the toe on one side of the leg. It can affect your whole arm or the whole side of the body. In addition, particularly in the genetic forms of dystonia, it'll affect much broader groups of muscles. In this case, this young lady is 19 years old at this time. Her entire back and head and neck musculature is not able to contract in the nice sequence that we are able to. This is the way she has to move around. This is the way she has had to move around from the time she was born. You can imagine this is not easy to live in this manner, okay? It's also a problem, not just because dystonia is not only genetic, it can be spontaneous as well. But dystonia is part of other problematic diseases, such as Parkinson's and epilepsy, making this a real problem to understand and disentangle from the experimental perspective. My interest in dystonia actually started long before I got interested in neural circuits. At the time, I was a fellow at Oxford studying the human genetics of these disorders. It was an exciting time because now one of my colleagues here, Dr. Huda Zogby, had cloned genes such as SCA1 for spinocerebellar ataxia, and the human genetics was moving forward. They then moved it to mice, where they could recapitulate the main problems that we see in the human patients. We had this great optimism that we'd have the same for dystonia and have these mouse models. Unfortunately, none of the mice ever had dystonia. They have some mild motor problems, but certainly not dystonia. I left the field for a good decade, sadly to come back and still no mice with dystonia. So a few years ago, I decided to take this on as a challenge and ask, what are we getting wrong by only understanding the genetics? Well, we went into the neural circuits here. I'm only showing you by DTI 2% of the circuits in the mouse brain. Already it's an overwhelming number of pathways. If I were to add the other 98%, it's a solid block of colors. We need to somehow go in there and disentangle what creates a dystonia, okay? We had some indications from some wonderful work from my colleagues, Bill Dower and others, Cameron Kodakar, who had started to appreciate that dystonia is not only a disease of a region called the basal ganglia, as had been thought for more than 30 years. It, in fact, is a disease of the whole motor system. And many parts of the brain have to come together to produce that very characteristic twisting and abnormal postures. What I was interested in was not necessarily what's happening in the ongoing motor system to produce it. I wanted to know what starts the dystonia. What triggers this devastating disease? Where does it come from? And can we identify that trigger point? My rationale was that if we know that, perhaps we'd be in a better place to start to understand how to stop the disease. Okay? So we then looked at this connection from the brainstem to the cerebellum, called the olivocerebellar network. We thought this was a great place to start. One, because it starts early in development. In mouse, these are already there during early embryogenesis, at least embryogenesis of the cerebellum. It's a very powerful connection. It's a one-to-one connection, which is very unique in the brain. Normally, you have many connections per cell when they go in there. Here, you have in magenta one afferent fiber twisted around its target, the Purkinje cell. But on its way, it makes 500 synaptic contacts, making this one of the most powerful, if not the most powerful, excitatory synapse in the entire brain. In addition, it has a functional signature. When we record electrophysiologically from these Purkinje cells and ask that Purkinje cells, what is that climbing fiber doing, it elicits a very specific action potential called the complex spike. So we can track them electrophysiologically at the single cell level, as well as anatomically at the one-to-one single cell level as well. The challenge then is that can we use this circuit model to create a dystonia? And here are some questions along the way we hope to address: structure, function, and potential therapy. What we did was hypothesize that we need to block communication between the synapse. Others had tried to lesion the olive, they've tried to cut the tract, with some success that you do produce a phenotype, and might I add, a very complex phenotype that includes dystonia and many other problems. The problem with the brain when you go tinkering and removing and lesioning and destroying, there's a lot of plasticity. And in the end, what you're analyzing in that context is not what you actually want to be analyzing. It's that plus many other things. We reasoned that perhaps we don't want to lesion or cut, but we want to block the way these two neurons will talk to one another. The way we did this was to come up with a genetic strategy to remove the ability of these synapses, the climbing fibers onto the Purkinje cell, to communicate through fast glutamatergic neurotransmission. The way we did this was used a genetic intersection approach and removed the ability of that climbing fiber. Oops, I'm sorry. I'm trying to go back. But it won't. Oh, it did. Okay, so we removed the ability of the climbing fiber to release glutamate. So everything's normal in the circuit except its ability to communicate by glutamate transmission. Here I'm showing you we've successfully removed a protein that's required for loading the glutamate into synaptic vesicles, and I'm showing you the electrophysiological trace here that we've selectively removed those complex spikes, the ones that are required for communication between these two cell types. Now, this would all be for nothing if these mice didn't have a phenotype. And when we were very excited to see that already at postnatal day 7, when we're trying to understand the development of it, you start to see postures that we thought were indicative of abnormal muscle contractions. 3 days later, you'll see this mouse is having a hard time righting itself, and paying attention particularly to the hind limbs, you see these severe hypercontractions, which we think were very much like a dystonia. When we let these animals live a little longer, it was even more striking, very similar to dystonia human patients. The more the animal tries to move, it throws itself into a dystonic attack, where you see severe contractions of the entire back muscles through the tail, as well as the limbs as well. Okay? We check this by using EMG to record muscle activity and make sure that we are actually getting co-contractions. When we look at muscles in a normal mouse, in order for, and the same for human, for our joints to move biomechanically in a smooth fashion, you need your muscles to move out of phase with one another. This does not happen in dystonia. Remember I said the muscles will work against each other. In this context, we were not too surprised at this point to see that the muscles don't have this out of phase contraction, but they ended up contracting together. But now we had a way of asking ourselves what is happening in the brain during dystonia. So we use electrophysiology to record these brain signals. And what you'll see, very similar to driving around Houston, if you're on the 610, things can be smooth going. But as I learned after moving to Houston, if you hit the 45 in any direction, communication between drivers falls apart, much like our system here. You get a lot of holes in communication, okay? But let's not blame the Houstonian driving alone. We need to think about the roads. Sometimes in Houston, it's almost like I was back in Africa with the potholes. Okay? If you look at the DTI imaging, we've segmented out just the outputs of the cerebellum, okay? And projecting out. Let's segment out what's happening in dystonia. So we think at this point, it's not only the communication, but also the pathways that this communication has to operate through are much reduced. So this bad information has to be channeled through less outputs as well. So there's a two-hit hypothesis potentially happening here. So what can we do? We started to think about how could we help these mice in terms of overcoming the dystonia. It struck us that perhaps deep brain stimulation could be one potential therapy. This requires you to put in an electrode in the brain and deliver a small current to try to repair some of that abnormal activity. This has been very effective for Parkinson's disease and tremor and some forms of dystonia as well. Much optimism for neuropsychiatric disorders. But in most of these cases, the cerebellum is not the target, and we wanted to test this. We think it's such a powerful connection to many regions of the brain, it should have a major impact on different regions. So we miniaturized the deep brain stimulation setup for mice, and I'll show you a small video here of, so this is a control mouse hooked up to a deep brain stimulation device. This is the dystonic mice also hooked up, okay? And you can see here, you've seen this video before where it's severely dystonic. Now, what my student will do here now is turn on the stimulator, and not more than 20 to 30 seconds later, that animal is able to walk around, it's able to groom and feed relatively normally. So just triggering trying to normalize the output, even though there's an incorrect pathway, is enough actually to give you much of that movement back. Now, I've told you that these can coexist. In dystonia, we think there's this kind of hiccupy type of activity. In tremor, you see this rhythmic problem. And some forms of ataxia, they're another movement disease, cells are firing much too quickly. We've had to segment out how this is happening, and we've been using optogenetic approach to stimulate the brain using a light-sensitive channel. And in doing so, we were able to ask ourselves and convince ourselves that there may be particular signatures for dystonia versus ataxia versus tremor. Okay? I'm just going to show you one example here independently in an otherwise normal mouse that we could elicit a dystonic attack without messing with anything, without anything else. Okay? We could also do the same for if we just ever so slightly tinker with this type of stimulation. Instead of producing a dystonia, we could now produce a tremor instead. Okay? So this is from the same pathway in the normal mouse. This was relevant, we thought, because as I've mentioned, if you have a disease that's complex, you may not have a pure dystonia, pure ataxia, or pure tremor itself. We injected this mouse with a drug called harmaline, which will induce the tremor. Okay? But what's happening here, we're using the mouse's own brain activity in a closed-loop approach, as well as the mouse's shakiness. What you can see is that there's an accelerometer feeding back onto the deep brain stimulation device. What we'll have now here is that every time the mouse shakes, the DBS turns on and stops the tremor. So we think this could be applied in a more complex fashion where you have different types of brain signals, different types of outcomes. The brain itself and the behavior of the animal will feed back on itself to turn on the deep brain stimulation in a closed-loop approach. Okay? I'm just going to finish off here by saying that we've used this rationale by trying to understand using drug therapies as well in a much more dedicated way, that we know what these drugs target, but it's the aspect of behavior we haven't appreciated. In this case, we use a drug called clozoxazone, which is a muscle relaxer, and we think it fills in those gaps, much like the deep brain stimulation. Okay? And here we have the electrophysiology. After treatment, it suddenly does. When these mice are on this little treadmill, that one fell off. It's the ataxic mouse. It barely hung on to the rotating device. After treatment, after a few days, we'll start in a second. It actually does a decent job of staying on. So we think we can use both the signatures of these different diseases to better the therapies, whether it's stimulation or make much more dedicated predictions of what drugs might work. Thank you very much, and thank you to my students, the support, and the NRI.
We have time for one quick question. Okay.
All right, thank you very much. Oh, so go ahead.
I thought it'd be interesting to give you the opportunity to ask the question that you would most like to ask.
Sorry, I didn't hear the last one.
So what remaining question would you like to answer for yourself?
I would like to see how what is it about these different diseases in terms of the circuit-based diseases? And we've heard about it today with epilepsy. You don't always see a morphological problem. That means embedded in that network, there's a neural problem as well. How similar are these problems? Because we see a lot of these gaps in the behavior of these cells, whether it's a dystonia, a tremor, and an ataxia. We actually see this in a model of epilepsy that we've developed as well. How similar are these different gaps, and how different are they? So that's one thing, and my motivation for that is to go back to the deep brain stimulation. How sensitive can we make these therapies to detect one set of abnormal activities versus another?
Okay.
Thank you.
Thank you.